Okay, welcome. I'm Tim Weaving, a PhD student at University College London, and I'm pleased to welcome you all to the 23rd instalment in Comp Biomed's e-seminar series in collaboration with the Virtual Physiological Human Institute, or VPHI. The talk today will be given by Mariano Vazquez, Chief Technical and Scientific Officer at ELEM Biotech, a startup company spun off from the Barcelona Supercomputing Center. Mariano has a PhD in computational physics from the Technical University of Barcelona, and his research interests are in cardiac computational modeling, computational mechanics, and high-performance computing. Before we begin, I'd like to briefly remind you that the session is being recorded and will be available on the Comp Biomed website and YouTube channel shortly. So please do use this to go back over parts of today's talks and others in the series, and then maybe even share these e-seminars with your friends and colleagues. There will be a Q&A session at the end of the talk today, so please save your questions until then. Then once the talk is finished, you can go ahead and post any questions you have in the questions tab, and then I'll be able to read them out for you to Mariano, and that way we can keep the session running smoothly. That's everything from me. I very much hope that you enjoy today's talk, and without further ado, I will hand over to you, Mariano.
Thank you, Tim. Thank you, Gavin, for organizing this. Thanks a lot. Thanks to everyone in the session. So, well, my talk is going to be about the supercomputer-based in silico clinical trials, which is the future of medicine. What is interesting, and I always try to stress, is that this is the future of medicine happening now. It's not just a future. It's something that it is happening now, and it is it is a very active field. Well, first I will give you an idea of where we are coming from. I like to present this like a pyramid. In the lowest layer of the pyramid, you have Marenostrum 4, which is this very large computer that we have in Barcelona. It's the computer in a chapel, as it is very well known. It's a very large one. It's almost 170,000 cores, 11 petaflops. This computer is being replaced. It will be a different computer by the end of the year, Marenostrum 5, which is going to be a 10 times larger computer, just closer to 0.2 exaflops. So this will be a pre-Exascale machine. At the next layer, you have the next level, you have the Barcelona Supercomputing Center, which is the research center that it is hosting the computer. So it's more than 600 researchers. It's a Spanish public center, and it belongs to Praise, and this is our new building. We have this building since last year. It is in the upper part of the city, close to the UPC, the Technical University of Barcelona campus. It was founded in 2005, and we're doing applied computational science since then. In those days, we created the ALIA development team. We started by developing by the team. This is the next level. ALIA is a code that we develop for multiphysics simulation. We started being 2. Now we are more than 50. Among them mathematicians, physicists, engineers, programmers of different backgrounds, and it was born at the same time of the center. And at the very top level of the pyramid, like the tip of the iceberg, is ELEM Biotech. ELEM Biotech is a spin-off company that we created 3 years ago, which is there to develop biomedical software technology. It's one of our code can be used in different verticals. So this company is there to commercialize and to bring the private sector into research and to transform this in a product to speed up the technology transfer for the vertical, which corresponds to the biomedical realm. What we do are in silico clinical trials. I would like to say this from the very beginning. We are not looking for just heroic, very large scale simulations. What we are doing is trying to use these large-scale simulations in order to create in silico clinical trials and run many of them. That's why it's very important to have a very efficient code that can run efficiently in these large computers. We are focused on organ level with contributions from tissue and cell. The technical challenge is really a challenge. In order to, the goal is to recreate these biological systems in a computer. Now, the more complex the system, the larger the computer you need. But in order to take profit of these computers, the larger the computer, the more efficient the code you need. And this is the technical challenge. If you really want to make these comprehensive, very large scale simulations, you need not just a computer, you need a code that is efficient enough to run in these computers. So our goal is to create this virtual human platform. Virtual human it's an avatar that represents a human, and in which you can test different therapies. It's like a human digital twin. So the mixture of these three things, like supercomputing efficiency, accurate multi-scale and multi-physics modeling, and virtual population generation, with this combination, you have these supercomputer-based in silico clinical trials. And the goal of this is to optimize and personalize the different therapies, medical therapies that can be used in healthcare. So if you create this platform, a user can follow these steps. The user can select the population, select the primary disease, select the comorbidities, then launch the in silico clinical trial on the selected virtual cohort. So it's running the simulations on for the different scenarios. Wait from days to weeks, let's say, depending on the tests. This is something that, in the case, if you replace an in silico clinical trial for a normal clinical trial, you have to wait years. So waiting days to weeks is very, very extremely reasonable. And then analyze the results as they come and repeat. Just to give you a bit of a background. ALIA is a parallel multi-scale simulation code. We started the code in 2005 also. So it is a more than 15 years old technology that was growing. It is used in different industry-related projects, like in aerospace, energy, environment, and the biomedical sector. And I'm going to talk about the biomedical sector. Regarding the biomedical, it is the only multi-physics, multi-scale code in such realm of this kind that was born and developed in a supercomputing center. So it is not that we are smarter than others. It's the fact that as the code started in such a center, we have these very stringent conditions for using the code, that it must be, and for developing the code, it must be very efficient in computational terms to use these systems. This, for instance, the movie that you're watching with some hiccups, but well, it's you can guess what's there. It's the one of the projects in which the code is used, which is not biomedical. It's a typical example of the massive use of the code, in this case in a wind field, and to simulate the windmills themselves. Just as a game, we started a few years ago to call everything that is used in which the code is used in the biomedical sector. We give the color red. So ALIA red is the goal of ALIA red is to do simulation tools for the biomedical research. So we basically work at organ and tissue level. These are not every problem in the biomedical sector, of course, require supercomputers, but where you require a supercomputer, that's where we are. We started with cardiovascular and respiratory. We're very focused in cardiovascular, but we're doing different things also. I will show some examples, like in a gallery of these two things. Our collaborators, but now customers, are pharmaceutical industry, medical devices industry, regulatory, CROs. So this is the vision when we talk about virtual human that we have. It's a vision that it is a it is the FDA vision that we completely share. It's this plot is widespread. It is very likely that you have seen it before. It's today, until today, every time that a pharmaceutical or medical device product pass the regulatory process, they have to pass these tests, laboratory, human, and animal tests, and with a bit of computer of any kind. But things are changing very rapidly, and now the scenario is becoming this one. You have less laboratory, human, and animal tests. Today, you still have them, but much less. You have much more computer-based testing, but also you have this new kid on the block, the virtual patient. These virtual patients represent a big complexity boost, because now you have to model not just the isolated therapy, you have to do it on the patient. So you need to model the system, the organ, the tissue, the cell. This implies a comprehensive modeling in which you have to consider different comorbidities, the patient variability. So this implies a very complex scenario, simulation scenario, in which you have all this variability. And then this requires specific in vivo and experimental validation, which is very different in biomedical than in other sectors, like, for instance, I don't know, aeronautics. You cannot put a strain gauge wherever you want to compare something. So you need to design from scratch a new way of validating, and this is done for different contexts of use. I will talk about this a bit later. So this validation stuff represents really a full different talk. It's a very important problem. So let me give you some examples in order for you to better understand what we are talking about. Well, first, the cardiovascular system. We have many collaborators. Important thing is not just to read the collaborators, but to see that the collaborators are coming from very different origins. They are academia, hospitals, companies. So it's very important to have doctors as your collaborators, because it's where the doctors take the reins of the research, and they pose the proper questions to be solved. Otherwise, you can really be distracted by the complexity of the problem itself. From the virtual, I will describe at a larger extent the virtual heart from the engineering point of view. So you have an electrophysiology model. Everything is solved within the same code, ALIA. So this is a multi-physics, multi-scale problem in which you have different physics governed by different sets of equations, all of them tightly coupled. So first of all, you have electrophysiology. In electrophysiology, the equations are diffusion equations with some nonlinear source terms, which are linking the different scales, the tissue model with the cell model. It's the nonlinear part of the problem represents the cell model. The cell model is in which the equations of the cell model, they govern the currents that are happening in the cells, in the myocardium cells. Then you have the mechanics coupled with the mechanics. So you have an electromechanical problem. In the mechanics is a nonlinear mechanics problem. It's dynamic problem also as the other one. It is tightly coupled with the electrophysiology through an electromechanical coupling, which is a problem in itself. It's the way in which the muscle develop stress from the electrical activity. So the electrical activity propagates, and this drives the mechanical contraction. And then after the mechanical contraction, there is a relaxation period, also governed by the electrophysiology. And next, there is the third physics that it is coupled in this problem, which is the fluid mechanics, is the blood. inside the vessels. The coupling is a fluid-solid interaction. It is more complex than the electromechanical coupling from the computational point of view, but it's a sorted out problem. It's not well, there are plenty of things that are published, but in this case the coupling is done, while in the electromechanical coupling the coupling is done at volume level at each of the Gauss points. In the fluid-solid interaction, this happens at the surface level, at the surface that are connecting the tissue with the blood flow. The blood flow is not turbulent, but it is highly transient in any case. So you need to solve all the scales. If you really want to study what's happening, you need to solve all the scales. In some, depending on the kind of problem that you would like to solve, you can consider as a Newtonian or non-Newtonian fluid mechanics problem. But then this is not all, because we are not working with a, as we always said, the nature is not a CAD user, so you cannot have the CAD of a person. So you need to somehow, at some point, include the anatomy and physiology of the patient in a way that typically you create this scenario from images, which is very, well, it has a complexity on its own. It's very difficult and also very artisanal. So you create your anatomical scenario and the physiology, which is also can be very controversial. And then with that, you have the scenario ready to run. But now that you run the scenario, the problem that you have is, well, you have to analyze the results, which are the results are not that simple to analyze neither. They are very large scale. They can be very large scale problems with small time steps, different time steps for each of the physics. And then you have to, and what is important is to analyze the results. You have to, it is not just the pressure or at a given point or the forces. No, because you need to validate and verify, which means that you have to compare with something. And when in the clinical experiments, even in vivo, in vitro, or with patients or real patients, you cannot measure whatever you want. So you need to measure biomarkers, and these biomarkers are specific complex quantities that you have to extract from both the computational and the clinical or experimental scenarios in order to compare. So validation and verification is a whole problem. So this is the thing with the in the biomedical realm, and that's why it's very different from the engineering point of view, because you have to this validation and verification is related to, as I said, the context of use, or which means what are you doing. So this validation and verification is not done. You do not validate your code. You validate your code under a given context of use that you define, and you measure the biomarkers, which are the quantities of interest. So what do you want to measure and what do you want to compare? And this is also, it's not trivial to establish this. So this is the general viewpoint from the engineering side of the virtual heart. So as you see, it's not just a complex multiphysics modeling. It's a complex multiphysics modeling wrapped around a very complex also scenario. Regarding validation and verification, it's like you have to follow some steps. First, you run a scientific verification in which your typically can be academic code self-verification in the sense of mesh convergence and time convergence. You can do analytic or manufacturer solution in order to check your code. Typically, this is done on very simple academic scenarios, maybe very small meshes in which you do this. The convergence analysis can go through very large meshes, because you want to converge and to see at what extent do you need a small mesh to capture all the physics and physiology of the problem. But this is kind of the first step. The next step is what we call the scientific validation. In the first one, you check that that your code is reproducing properly the equations that you want, and the next one is that the equations that you want and your code are reproducing at some point reality or getting closer to reality. So you do this scientific validation, in which typically you do frequently there are simple cases coming from literature and mostly single physics. But then there comes these two different validations, which are and for the first two ones, it is a continuum validation that you do, or every time that you develop something, you try to validate. But in the green and red, in the experimental validation and clinical validation, you do these 4 campaigns, because in this case, for the experimental validation, for instance, you do benchtop, ad hoc benchtop in vivo or ex vivo experiments, which is very difficult. They are very, very difficult to set up, or could be very difficult to set up. You have to follow a very stringent validation protocol. You need a high control, and maybe it is very frequently it's a multi-physics problem, and the best outcome is to have some kind of certification. In this case, for instance, I will show you some example later. You compare your code against ad hoc benchtop experiments done in a lab, in which you have a full control of the experiments. And then the clinical validation, which is kind of the top of the top. It's in this case are clinical in vivo measurements. So you have to follow a clinical validation protocol that you have to define, and then you have to ask your colleagues to believe. In this case, you have low control. Typically, the data typically is not good. It's not at all that accurate, because it is coming from patients, and very likely it's going to be a multi-physics problem. So the best outcome that you can expect from this, there is no certification for these sort of things. So the best outcome is a clinical validation paper, in which you can, with these 2 last ones, you can really have the confidence of your colleagues and the potential users of your tool that you are doing something that is relevant. And you are getting, in this last one, is you can say that you're getting closer to the virtual patient, a patient that represents virtually the reality. So this is what we do with the virtual heart. You have a patient's database. We have a patient database with different high definition images from explanted hearts from female, male, and children. In this case, we are working with the Visible Heart Lab, the University of Minnesota, but we're also working with other partners like UCL, for instance, for having these patients database. Once you have this patients database, what we have, it's a way of transforming these. In this case, for the Visible Heart Lab, it's about 40, 50 real patients, by doing some morphing and transforming from one moving continuously from one geometry or one patient to the other. We can have thousands of patients, and we can create this virtual patients database, again with female, male, and children, with which we can analyze hearts in health, in disease, and to improve the treatments and therapies for them. The anatomy and physiology is very complex in order to setting up the scenario, and this is just I wanted just to show you one example. In a heart, what is very important is the anatomy of the heart, in order to have a good definition and a good simulation scenario. So you need to define the muscular structure of the heart, which is not simple, and it is information that you cannot get. It is very likely that you cannot get, not at all, from a living patient. So you need to do something in order to have a anatomical structure. So there are some semi-empirical rule-based models in which you can create this structure and then use it. This is the fact is that the tissue is like a composite material, in which you have an active part and a passive part, and the active part and it's fully determined by these fibers. They are like, it's like a composite. So you have these fibers which are embedded in a matrix. I will show you some examples. This one is an example. This is something that we are doing in pharmaceutical industry. So what we are doing here is to analyze a cardiac safety assessment. So in here, what we do is to, when a drug arrives to the market, you can have some problems related to the cardiac security. Eventually, you can have cardiac arrhythmia, and that's what we are studying, to study this the potential for cardiac arrhythmia. So we have a risk, a measure of a way of measuring the risk in resting condition, and a way of analyzing the arrhythmias that can appear in a stress test. This is just some picture. It's very technical, but the important thing here is to capture the stiffness of these plots. So in gray, you have the experimental. The important thing is the stiffness here. So in gray, you have the experimental one, and in color, you have in the bluish and brown color, you have what we have for different for different patients. So you see that we have more or less the same stiffness. What is this stiffness meaning? This stiffness means how the cardiac risk augments when you increase the dosage. So what you have to do here is to set up your population, female and male. You set up your population. You set up the inputs that are defining the drug and the drug action, and then you define the range of the dosage. And with that, you can study the stiffness, and you can study the statistical distribution of the patients. And with that, we can see that our model is capturing the stiffness of these two drugs. We have done this also for other drugs, and we have these results which are very encouraging. This is the typical thing that you see when you provoke an arrhythmia when one of these drugs can provoke an arrhythmia. This is a typical case of a drug that was delivered and administered for during the COVID pandemic for COVID disease people, which is called hydroxychloroquine. It is very likely that you have heard about it, and you have heard also that this drug can bring cardiac problems. This is a cardiac problem, an arrhythmia, which is called this left bundle branch block, and that naturally appears when for some dosages of this drug. So here it is not just a risk of arrhythmia, but we capture the arrhythmia itself. This is an example of a problem in which this is more for medical devices. So here at the center, the movie that you're seeing, what you're seeing it is the two ventricles that are contracting. The atria there are just for mechanical purposes. They are inert, but electrically inert, but they are there for these mechanical purposes in order to make the heart move properly. So this is colored by, the tissue is colored by the electrical activity, which drives the contraction of the ventricles, and the ventricles are pumping out the blood flow. We are interested at the systole period, which is the period in which the heart contracts. This is a healthy heart. Now, the one that is activating now is disease heart, in which they have a problem at the like it is left bundle branch block. It is in the left ventricle, but it is in the image is the ventricle to the right, in which you have this conduction problem, and we have placed here a pacemaker in the flash that you see to the very right. So we disease the heart and we place a pacemaker, and then we analyze, like in this table, we can analyze the heart in the healthy situation, in the disease situation, and with different synchronization periods for the pacemaker. What we are analyzing here is the some strokes, stroke volumes, like the left ventricle ejection fraction and the left ventricle end of systole volume. So what we want to see is that we can recover the cardiac function by placing somewhere and changing the places in which the pacemaker is seated, and also the synchronization. This is another example. This is another pacemaker. This is a much more sophisticated one, in which you see it's this the thing that you see here, the kind of a small cylinder pin inside the ventricle. So this is all the pacemaker, the cylinder. And what the collaborator wanted to see here is the forces on this pacemaker, because of course you don't want that this pacemaker just get loose in your system. So the forces in this pacemaker and the forces in the muscle itself, which is the stresses are colored in the movie at the top right. And also something that is very important for the medical devices is the potential to produce thrombi. And we study you can study this by analyzing some flow patterns, that like the bluish ones that you see here, which is the Q criterion, is a way of measuring the vortices and the potential for producing thrombi in the heart. And finally, of course, what you want to see is where do you have to put the pacemaker and how to synchronize it to have this to recover this cardiac function. This is another test, that another problem that we are solving. This is a clinical validation that we are doing with St. Paul Hospital, in which we have this information from patients. In this case, you see that the heart is not complete, because this is the best way, the best thing that you can expect from real patients. These are the kind of images that you have and you can measure. So in this case, what we are doing is we are analyzing patients that have already went through, this is retrospective data, patients that already went through therapy based on pacemakers. So we study what's happening before the pacemaker implant and after the pacemaker implant. And we study, this is what it is called the acute scenario. So just right after and right before. So we analyze different biomarkers. This is electrophysiology, electromechanical problem in which it is electrophysiology is the one that is colored and that you see. And also the contraction of the heart. We can analyze not just electrophysiology, things like electrocardiograms, but also the ejection fraction of the heart, which is the power of the heart to pump the blood. And we can study these things, as I said, just right before and right after the implantation of the pacemaker. This is the typical example in which you compare now with experimental results. This is something which is called the left ventricular assist device, which is a pump that it is used for, external pump that it is used for people with have a very advanced heart failure condition, so they are ready for a cardiac transplant. So while they are waiting for, it is very likely that you need this pump to assist the pumping function of the heart. So this is an experimental measurements. It's an experimental arrangement in which our collaborator in San Diego State University, they have this experiment and they can measure different things in it, and we compare what they have with what we have. But we do it like in a clinical trial, but running many patients under different conditions, and we measure the sensitivity matrix, and we can produce the sensitivity matrix and can analyze that statistically we can have the same behavior as the experiment. It's a very nice one. In this case, what we are studying are the valve mechanics. what you see here, these two movies, is studying different conditions for a valve. So the one to the left is stiffer than the normal one, and the one to the right is softer than the normal one, the tissue material. And we study what we wanted to study here is the vortex production and the complexity of the flow downwaters. The idea here is to study the capacity of production of thrombogenicity of the model. And this is the study. This is like it is not placed in a heart. It is also trying to follow an experiment. So it's like a virtual experiment that it is produced with this and measured with this simulation. With this, we can check different. It's like creating a virtual lab for these valve implants. We can not just to, well, we can compute these flow biomarkers for thrombogenesis, and also we can check the leaking under the different flow condition, the position, and also the materials for the cusps, the angle, the size, many different conditions. And with that, we can create many patients and study the statistical tendencies. So this is the sort of interface that we can design for this problem. This is just a glimpse of the sort of interface. The idea of this interface is try to eliminate all the computational complexity and leave all the physiological complexity and the statistical analysis complexity to set up the trial and analyze the results. We are developing this interface. Now, about to finish as the last example, I will just give you one example of the respiratory system. Again, you see those collaborators which are from different places. And in this case, this is a patient coming from our colleague at Imperial College London. So they have this patient geometry, and with this patient geometry, we run particles. These are Lagrangian particles that are computed on the fly in order to have all the accuracy as possible. So this is not post-process; it is computed on the fly. And as you probably know, the paradigm for parallelization of particles is very different than that of continuum mechanics. So it's and if you want to do this efficiently in parallel, all in the same code, you have to take care of many technical problems. So the goal of this case is to elaborate a deposition map for the particles. Depending on the size of the particle, the particles are sitting at different parts of the system. Some of them at the upper airways, some of them at the lower part of the airways, depending on the size, on the position when you start at the inhaler, where they are getting out of the inhaler. So this is the kind of this is very useful for studying different inhalers and nebulizers and try to establish the optimal functioning condition, and also including the patient variability. And I like to show this one because it is very beautiful and it is also very original. It's something that we are doing. We changed the color now. It's not just ALIA red, it's ALIA purple, and it's the virtual pregnancy lab. So what we're doing this with Washington State University in St. Louis, and it's pregnant uterus simulation. Well, the uterus is a muscle, not exactly as the heart, but very similar. So we can fine-tune our tissue model in order to make it closer, to make it behave closer to the uterus. So there are some pictures here on the comparison of these preliminary results experimental measures taken in women, and it is very encouraging, because what we are having our simulation is very close to what it is observed, and we are very happy of this. These are very complex simulations, but very nice, and also with a powerful application. So our vision is that what we would like to have is a human digital twin with our virtual humans. We are solving different systems of the heart, and at a certain point, we would like to, in the ages to come, to make better modeling for each of the different systems, and finally link them all together to create this human digital twin. So as a conclusion and future lines, it's well, as I said before, high performance computing is not needed everywhere in the biomedical realms, but for some problems, which are many problems, it is the only way. And this is getting larger and larger, and that's why we need computers that are more powerful and codes that are more efficient. But the fact is that HPC is mature enough to be used. You have plenty of algorithms, solution schemes, implementation strategies, the mathematical models. And then from the purely computational point of view, you have load balancing techniques in order to make this happen more efficient way. You have different programming models. Hardware now is much more available because, well, computers are everywhere, large computers are everywhere, and they are available to be used. Software also, like ours, more input data sets. So this is not science fiction. This is something that it is happening now, and this is the important message I would like to convey. Some of these simulations can look like science fiction, but they are not at all. In fact, I can tell you there are companies that are paying for this. So if a company is paying, then it means that it's useful for them. And okay, but a lot must to be done to increase the confidence in these tools. But well, look at other fields now, like aerospace, energy, climate. In all these fields, computer models are at the core business. So why not in biomedical industry? So both industry and government have to fuel the pace. So it's very important to learn the COVID-19 lessons, that when you have these sort of tools active and massively used, you can have plenty of benefits. But well, more hands are needed, especially on the clinical and medical insight, because we need to, and this is like a virtuous circle. If we can bring more clinical and medical insights in our tools are going to be better, and this will bring more clinical and medical insight. And I think that this is all on my side. So thanks a lot for your attention.
Super, thank you very much, Mariano. Just a quick reminder for the audience that if you do have any questions, now's the time when you can go ahead and write them in the questions tab so that I can read them out for you. So whilst you all gather your thoughts, I thought I'd ask you a few questions of my own, Mariano. So first of all, how are these models validated?
Well, as I said, it is indeed we need more a couple of full talks about this. I have given a glimpse on how to validate, but the importance in validating the models. Well, first of all, on how to validate. Well, it is something that it is being established. There is no defined way of validating these tools. So it is you must be very creative in the way of doing it. And it is, once you run many cases, then you have to, when all the results on the table, you need to establish your own strategy to validate, and you want to contrast this strategy with your colleagues. So it's very difficult. Now, this is the how to. And now, why is this important? Because this validation above the obvious, that it is to validate that what you are doing is okay and represents really nature, is that in this validation goes the confidence of the users. So if you really want to transfer this technology and make it translational and move it to the medical realm, then you cannot just show that you're validating. You need to somehow bring the confidence of the users in your tools, and this is that's why validation is very important, and because of it is a credibility matter. Now, given that that validation is so complex, well, it's very creative, and it is something that it is being developed. It's being developed.
Sure. I mean, I suppose we kind of saw it on one of the slides there, right, where these complex models, they can be very sensitive to the initial conditions, right?
Yes,
sure.
yes, and also in the initial conditions initial conditions are real because it is the variability of the patient. So it is not that you do just to test to measure the sensitivity. No, it is real. The patients are really very different, and that's why it's decisive. Yes.
Sure, absolutely. I had a question kind of related to the patients, actually. I just wanted to check if I've understood this correctly. So I think I recall from one of the slides, you kind of start with this core group of real patients, right? And then you spoke about effectively like extrapolating to a larger group of virtual patients. Am I understanding it correctly that you kind of extrapolate out from the real patients, or you have this kind of database of virtual patients that you just
this is a very
the sample with?
Yes, this is a very interesting point, and it is a very important question, because the fact is that you cannot just interpolate. Let us suppose that you have one patient, two different patients, and these two different patients represent different physiological models for the inputs and different anatomical models. You cannot just interpolate between one and the other, because it is dangerous, because perhaps you can have some patients, virtual patients, by just purely interpolation, the pure interpolation, that you can have some patients that are not realizable, that are not real. So you cannot just vary at random the properties in order to have an environment around one patient. So you need to do this in retaining the physiological meaning. So, for instance, we have patented one way of doing it, because it is not simple. It is not just interpolating or just varying randomly the properties. So it is difficult, and it is the way we do it is, well, you apply some morphine, but keeping the physiological meaning. You have some ways of defining the properties. The input properties are not, you not just define variation ranges for the properties themselves, for the inputs themselves, but for combinations of the inputs. So it's very artisanal and very difficult. Yeah.
Of course, thank you for that answer. I can see that we've got a question through from the audience here. So Els Disvert asks, is modeling feasible through a cloud solution, or do we need HPC and parallel computing? And is it feasible to deliver HPC through the cloud as well?
Very good point. If you have asked me this question about 3-4 years ago, I would say no cloud. But today, I can confidently say, because we are doing it, we are using it, yes, cloud computing is indeed a very good solution. Why I'm telling you this? Because the models that we are using for solving, for instance, I can give you examples. In the case of the pacemaker that we are doing with Sant Pau Hospital here in Barcelona for placing the pacemaker, these models are run in a few hours using 500 cores. In the case of the drugs, same case, few hours using up to 2,000 cores. So today, cloud providers can give you instances of 2,000 cores that can work almost as efficiently as a bare metal solution. So yes, you can deploy this on the cloud. This is one side of the answer. The other side of the answer is that centers, like HPC centers, like for instance EPCC or also BSC and also SARA in the Netherlands, they give you access like a cloud to their high performance computing solutions. So the important message to convey is that we are arriving at a point at which in the cloud you can have the instances that you need to solve these sort of problems, and that this is something that it is one of the reasons why this is coming out from science fiction and getting closer to the clinical realm.
Sure, I see. Another question from Els Desvert here. Would you like to deliver services to hospital teams, medical doctors, or even even patients themselves?
This is very interesting because we are not doing typically you have two, or let's say the traditional way of facing these things about 10 years ago is that you say that what you're doing is patient specific stuff. This is one way of presenting things. The other way is saying that you are not at all doing patient specific. We are not doing patient specific. All that I showed you here is not patient specific. On the other hand, what we are doing is, let's say, population specific. So what we are doing is we are creating a population and run a simulation on the population, but we cannot say at this moment that one of these patients is representing exactly a given patient, but it is representing one patient, which is not a given one, let's say, with name and surname. So something that if the doctors, so now answering directly the question with this as a premise, what we are doing now is trying to, first of all, to present this to the doctors. So the doctors are going to be those that are encouraging, key opinion leaders that encourage the use of it, and giving some free access to the doctors, to some selected doctors to use it for them to let them know the solution, and at the same time working with pharmaceutical companies, medical devices companies, regulatory bodies. So the goal is that today it is like this way, but the final goal is that in the future, in the next, I don't know, maybe 3, 4, 5 years, the doctors themselves, now at this point, they will use this to help them in the therapy for a given patient. And why not? That the patient himself or herself can, if not run the simulation and set up the simulation, but watch at his own and analyze his own simulation. We think that this could be a very interesting way of putting medicine in the hands of the patient itself, in order to check with very highly detailed his own physiology and his own body. But we think that this will come, let's say, in 5 years' time.
Absolutely perfect. And a follow-up to that is, what data do you actually need to construct a virtual twin of a patient?
It is like we are getting there. So one of the goals of we have this vision that finally we can solve a given patient. We can simulate a given patient. This is our vision, and going towards this vision means that we are kind of grabbing data and curating the data in order to identify which are the data that we need for simulating one patient. So as you say, Tim, we are just getting there. We are trying to establish which are the data that we will need to model a given patient. So we are getting close to that.
Sure, and you said that even within 5 years or so, you reckon we could start seeing actual realizations of this,
yes.
right?
Yes,
Amazing.
it is. it will not be out of a sudden. It will be something progressive.
Sure.
Because, well, it is something intuitive in the sense that if you are modeling a population, at some point you will identify persons, singular persons within this population. Why not? So by modeling populations, we are getting more and more knowledge on how the individuals are can be modeled. So we think that progressively in the next years we are going to come close to the point at which we can say that we are doing really patient-specific stuff. This will come.
Amazing. Yeah, I remember hearing of personalized healthcare maybe 4 years ago and thinking, you know, that's so far off. But it's amazing that you say it's quite a lot, maybe seen a lot sooner than I might have expected.
Yes, at least for the kind of problems that we are solving. I mean, I know that, well, we'll know or probably know that, for instance, for hip implants or prosthesis for knees. In this case, the modeling and simulation is getting much closer to the patient. But for the kind of simulations that you have seen today, no, we need to wait for a few years more.
Of course. Okay. Well, I'm not seeing any further questions coming in from the audience, and that was everything that I wanted to ask as well. So thank you once again, Mariano, for giving us your time today. I thoroughly enjoyed the talk. I'm sure the audience did as well. Just a reminder that this e-seminar will be available on the CompBiomed YouTube channel and website shortly. So Mariano, if you wouldn't mind going on to the next slide, that is accessible via the link shown on screen here. But just before we end, I'd just like to take a moment to plug CompBiomed's free scalability service as well. So if you're someone who uses or even develops biomedical applications that necessitate intensive computation, but maybe you're running into performance issues that are limiting the scope of your research, then CompBiomed offers a service completely free of charge to improve the turnaround of your computing jobs. And Mariano, if you wouldn't mind going to the next slide for me. So if this is something that applies to you and your research, then you can fill out an application form available on the webpage displayed at the top of this screen. Otherwise, please feel free to reach out either at the email address shown here or via the In Silico World Slack channel. Next slide, please. So In Silico World is a community devoted exclusively to in silico medicine, offering expertise, opportunities for collaboration, and a safe space to exchange ideas or advice. And next slide please, by the way. Already, In Silico World has a large following, including members from large biomedical companies, biomedical SMEs, independent software vendors, clinical research institutions, and even regulatory and standardization bodies such as the FDA and NICE. So that I just wanted to very briefly mention that. And finally, thank you to everyone in the audience for attending today. I hope that you all enjoy the rest of your days. And thank you again, Mariano.
Thank you guys. Bye-bye.